So all of us, knowingly or unknowingly, use a lot of nanomaterials. For example, in sunscreens, in separation or water filtration, and also in industrial cases. But when we look at the manufacturing of these nanomaterials, a clear issue comes out, which is the amount of waste created when producing these nanomaterials. An analysis has shown that when we want to make 1 kilogram of nanomaterial, between several thousands to 1 million kilograms of waste is produced. And there are several reasons why this amount of waste is created. And these reasons include the need for high purity feedstock, which requires purification and high energy inputs. It also comes to low yields of production, so only between 5 to 10% of the starting material ends up into the product. The requirement for sequential processing, which means that at each step or at each sequence we are producing waste and it simply gets multiplied. In some cases we have toxic chemicals being used or toxic or hazardous byproducts being produced which contaminate the waste. And also need for specialized environments such as high vacuum or high temperatures or long duration at high temperatures. And all these cases require high amount of potable water and high amount of energy inputs, which we could have otherwise used for sustaining human life.
So how you make nanomaterials is, as chemists, we tend to use a wet chemistry method, which is where you get two reagents that are dissolved in solution. So we call that soluble. And when we add them together, then the mineral we make, the nanomaterial we make, falls out of solution. So it becomes insoluble, and then we can just simply filter that off and we have our mineral. The problem is when you do this in ambient conditions in room temperature, you get a huge variation on the shapes and the sizes, and the quality of the material is very poor. So what we do is we look to be inspired from nature. So nature does a lot of biomineralisation. You will be completely aware that your body's got a skeleton and teeth, and these are obvious minerals that your body has biomineralised. Similarly, if you think about sea creatures, they make shells around them. So there's a lot of biomineralisation that goes on. The examples I've given of on kind of a large scale, a macro scale we call, which is like meters to 10 centimetres in scale. However, we look and we see that nature also biomineralises at the nanoscale.
So one of the examples we're looking at in terms of materials is silica, which is SiO2. And there are microalgae called diatoms, which grow in ponds and also in drinking water sometimes. They produce biological silica. So diatoms selectively uptake the precursor for silica, which is essentially dissolved rocks in any water environments. It then uptakes that and concentrates it within the cell, and those concentrated pools of the precursor are taken to the site of silica deposition. Now, at the site of the silica deposition, there are proteins and other organic molecules which pre-assemble into the final form, and as the precursor arrives there, it catalyzes the formation and also the templates the formation of biological silica, thereby making a cell wall, which is what the diatoms use for their protection.
Magnetic bacteria are a few micrometers long. A micrometer is 1/1000th of a millimeter, so they're very small. And these are small, spiral-shaped, like little worms, if you like, the bacteria, and they swim around in watercourses, so rivers and seas. And what they do is they take up soluble iron, so there's always a bit of dissolved iron in the environment. They take up that dissolved iron and they biomineralize magnetite nanoparticles. And how they do that is that the dissolved iron, the soluble iron, goes through their cell and it gets concentrated into a lipid vesicle, which is like a round hollow membrane called the magnetosome vesicle. And in that vesicle, there are proteins that are gathered together and aggregated within this on the inner surface. And these proteins are specially designed to nucleate, so like basically start the formation of this magnetite mineral. And there's also proteins there designed to control the shape and size of them. These proteins that are responsible for forming the particles within the magnetic bacteria. To understand how they do that, the best way we can think of doing it is to take those proteins outside of the cell. And what we found, which is really interesting, is that when you add this purified protein to a simple chemical precipitation of magnetite, if we add the protein, a specific protein from the bacteria, to that reaction, we get really nicely sized and shaped particles. We can see that actually there's different proteins will control a different shape. And some actually will only nucleate magnetite and really control tightly the mineral that's formed, while some control the shape. So now we've got this understanding of the different things that control the nucleation of the mineral magnetite. We can use that understanding to now try to make cheaper, more easily accessible synthetic molecules. So all we need is those groups that give that negative surface for the nucleation, or the positive groups organised in the right way to bind to magnetite surfaces. That's all we need. So why don't we try to mimic that in a more simple, easier to produce molecule?
So in terms of scaling up, a lot of people don't realize that scale up is not trivial. So it's not simply taking it into a large reactor and mixing it by hand or mixing it by a standard mixer to get the products. And the reason for that is the self-assembly of all these additives and organic molecules, as well as the entire particle formation process, is completely affected by how well things are mixed. What are the flow conditions, whether it is a turbulent flow or a laminar flow. They all determine, first of all, the chemistry of the reaction, how complete is the reaction will be. But second of all, also the particle formation, how large the particles will form, how will they aggregate, and what will be their final shape. So as you can see from the illustration is that the self-assembly and particle formation is different when you have no flow or where you have really hand-based mixing at small scale compared to the flow in a model system or flow in a real large-scale reactors. And that has a significant effect on how the self-assembly and product formation takes place.
What we need to find out is how the additives, which are the biologically inspired molecules, aggregate together and behave in numerous different situations. And we can do that through computational modelling.
Modeling means that we take nature and we simplify it in such a way that the most important properties are still there, but the less relevant properties are taken out. So, for example, a model can consist of a number of equations. And typically they are too difficult to solve on paper. So basically you program the computer, you write a code, and then the computer solves the equations for you. And this is then called a computer simulation.
So what we have seen so far is discovery and implementation of a green or sustainable manufacturing method for nanomaterials. And this method offers significant advantages, such as it is really a fast process. It occurs at room temperature, it occurs in water, it occurs at ambient pressure, so does not require high or low pressures. And the unreacted material has the potential to be recycled. That includes the feedstock and also the organic additives. All these benefits come together, actually compound in terms of providing huge amount of potential in terms of making cost-effective yet high-value nanomaterials.